Hello everyone, welcome to our new chapter. This is going to be chapter 4, which is going to be all about proteins. We're going to take a look at their structure, at their function, and we're going to do so in a 3-part series. So our first part, we're going to go ahead and take a look at exactly what a protein is made out of and all the processes that it has to go to fold in its formal shape so that it can correspond with the function that it needs to complete. Now, the entire chapter is basically broken down into 4 main topics. The shape and the structure of proteins, as I mentioned to you before, that will be discussed in part 1. And then when you listen to part 2, we'll talk about how proteins actually work. And then part 3 will combine 2 of these topics. It'll take a look at how proteins are controlled and also what are some of the methods that researchers utilize to study these proteins so that we can learn about all these amazing facts and features that they contribute to our day-to-day life. Now, speaking of functions for the protein, it turns out that there are a lot of them. Proteins are often described as the workhorse of the cell, and that is because they have a hand in numerous different functions. In fact, if we take a look at the mass of a cell, or I should say the dry mass of the contents of a cell, we see that proteins make up the majority of that weight. And that kind of makes sense. So for instance, you can take a look at panel 4.1, which is illustrated on the slide. If you take a look at the massive amounts of functions that proteins are able to provide, it makes sense why they play such a huge role and are found in such large quantities within an individual cell. So we could start off with something as simple as an enzyme. Enzymes you've probably heard about in your different classes. Enzymes are described as biocatalysts, meaning that they can speed up chemical reactions. Oftentimes they do so by lowering the amount of energy that's required. This is amazing for the cell because that means they can get the reaction done quicker and get to their products at a faster rate. Proteins will also play a role in structural role, giving mechanical support to cells, to tissues, to organs. If you look at the illustration for the second box, you notice that they have hair, and that's because proteins like keratin are responsible for a lot of the features that we have when we take a look at our hair strands. Is it straight? Is it curly? Proteins also play a role in transportation. So for instance, our red blood cells are filled with hemoglobin, which is a protein-based molecule. That hemoglobin allows oxygen molecules to attach to the red blood cells so that it can be transported throughout the rest of the body. How about something like movement itself? Well, proteins play a role in that as well. We have motor proteins, so things like actin and myosin that play a role in the formation of our skeletal muscle, for instance. They are the ones that have to interact to make cross bridges, do things like the sliding filament theory so that our muscles can successfully contract and relax. We also see that proteins can play a role in storage. So for instance, they'll help us store iron in the form of ferritin. We know that amino acids can be found in egg white. That's another way that the embryo can utilize some of its food mechanisms. So it will store, the amino acids will be stored in the egg white while the embryo continues to develop. Beyond that, we have signal proteins. These are able to allow for signals to be relayed, whether it is a protein that is on the outside of the cell, communicating with the inside, maybe through the use of channels or receptors or activation of like secondary messengers. The formation of channels itself, those tend to be done by proteins as well. So that way we can do things like diffusion, osmosis, active transport, any type of mechanism to move stuff in and out of the cell usually involves a protein at either a minute or a major level. We have receptor proteins, which are able to detect a signal and cause a response. We have our transcription regulators. Those are actually very important in order for our genes to be activated, meaning that they initiate transcription and eventually translation. So to go from DNA to RNA to proteins, we rely on transcription factors to initiate everything. And those are proteins that basically regulate when the genes will turn on and off. And even with everything that I've mentioned so far, we also have proteins that are what we like to call special purpose. And these are proteins that are going to, for instance, play a role in the formation of hormones, in the formation of antibodies, just such a wide variety of different functions that these proteins actually do. So it comes as no surprise that this macromolecule would get an entire chapter dedicated to its shape, its structure, and overall to its different functions that it can play a role into. Now, first things first, please remind yourself that, as we discussed in one of our previous chapters, that a protein is a macromolecule, and it's composed of subunits called amino acids. So the amino acid is going to be your monomer. And when you look at the general shape of an amino acid, what we see happening is that they will always have a central carbon in the middle, and that central carbon is going to be bound to 4 different items, okay? One side will be bound to a nitrogen and 3 hydrogens, and this is the part where you have to forgive me because I can't draw fast enough with my little mouse. The side that has the nitrogen with the 3 hydrogens, the NH3, this side is going to be called our amino group. Okay, see it right over here. And as you can see in our ball and stick model, the nitrogen is using the color code of blue, and then the hydrogens are color coded as your white. Over here in black, that's going to be your carbon. So let's come back to the one I drew. What we see happening is on the opposite end of the amino group, you're going to have your carboxyl group. And your carboxyl group is consistent of another carbon, and on one side is going to be double bonded to an oxygen molecule and also bonded to a hydroxide molecule, which is an O and an H. So they like to write it COOH. Okay, now this side of the amino acid, that's your carboxyl group, and this side of the amino acid is going to be your amino group. You're going to hear me mention those terms quite a bit, so make sure that you know that they're just either sides from the central carbon, and that all amino acids will have an amino group and they will have a carboxyl group. We've discussed these before in our previous chapter when we were going through the process of dehydration or condensation and hydrolysis. And we talked about the fact that the amino group has to give off its hydrogen and the carboxyl group has to get rid of the OH, its hydroxide group, so that the amino acids can form together, can bind together, and create their peptide bond. And if you need a little reminder, that's perfectly fine. You can take a look at this illustration right here where you can see that you have your glycine amino acid and your alanine. And if you want to combine them together, you're going to have to do dehydration. See how the water molecule is removed? And that is going to be done through the fact that we are going to remove the hydrogen from your amino group and a hydroxide from the carboxyl group so that they both have an opportunity to bind together, and that bond that they make when they link together, when you link the glycine and the alanine together, that is an example of a specialized covalent bond called a peptide bond. And that's one of the reasons why when you link amino acids together, you create a polypeptide. And as the polypeptide chain keeps getting longer and longer and longer, it starts folding, and that will then make your way to the ultimate shape of what a protein is needed for. Now, my apologies, I left my little drawing astray. So let's go back to my little drawing of a typical amino acid. We have the carbon in the middle, we have our amino on one side and our carboxyl on the other, and the other 2 binding sites for the central carbon, one will just be a simple hydrogen, okay? And then the other one is what we're going to call our side chain. And the side chain is often represented by the letter R. And what we see happening is that there's 20 common amino acids that are out there. And if you ever want to pull up the chemical structure of all 20 amino acids, you're going to notice that all 20 of them have the central carbon, they have the amino group, they have the carboxyl group, they have the hydrogen. And what sets them apart, what makes them distinguished from their other 19 friends, is whatever we put right here in the side chain. So the side chain is where all the variety is going to occur. So for instance, if I'm coming back over to my drawing, I'm going to notice that for glycine, for the side chain, that just means that I added an extra hydrogen, and that makes my glycine. On the other hand, if I come over to my alanine, I see that my central carbon, which is this one right here, I see my amino group, I see my carboxyl group, I see my hydrogen. So that means that this part right here is my side chain, and it turns out that black is for the carbon, and then remember the white is for the hydrogen. So that means that in the side chain of alanine, we added 1 carbon and 3 hydrogen, and that's what makes it unique for alanine. So all 20 amino acids will have 3 similar sides with the free hydrogen, the amino, and the carboxyl group, and what will set them apart is whatever we put in that particular side chain. Now, when you start linking together your amino acids, always keep in mind the whole we're removing a hydrogen from the amino group and a hydroxide from the carboxyl group, and where we link them together, that's going to make our peptide bonds. And peptide bonds, as I mentioned to you before, they are a specialized covalent bond that you exclusively will find with the interaction of your amino acids. When the amino acids link together and they form their peptide bond, one thing you're going to notice is that you will have a section of what we like to call the polypeptide backbone. And the backbone here featured in the gray boxes, notice how they're lined up together. They're making a very uniform pattern, and that is because this is the repeating structures of that's being recognized within each amino acid. So the fact that they have the central carbon, the hydrogen, the amino group on one side, and the carboxyl group on the other end. In fact, something that I want to point out to you as well is when you look at your polypeptide backbone, notice how on the left-hand side, you're going to have your amino group that's facing towards the beginning, and on the right-hand side, you have your carboxyl group that's facing towards the end. This is the typical format of reading your polypeptide sequence, meaning that when you line them up, you start by reading the amino, or you should start by facing the amino towards the left, and then you read towards the right-hand side. That is where your carboxyl group is going to be featured. Stepping away from the polypeptide backbone with its repeating subunits, you're going to notice that they've also used different color codes. That's going to be, for instance, here in blue, we have our yellow, we have our green, and we have our pink. And notice how those have all been marked as side chains. So this is what's giving that unique characteristic to the different amino acids that have been linked together. The side chains are what's going to allow for the amino acids to be chemically distinctive and obtain their very unique qualities that we come across. So once again, the side chain is what I featured on our previous slide as labeling it by the section R. So lots of variety you could see. Here's your histidine, aspartic acid, your leucine, and your tyrosine. And the colored sections, the pink, the green, the blue, and the yellow, once again, that's going to be our side chain. That's where the distinctive property is going to come from that will set one amino acid apart from the other 19 that we are known to play with. In fact, here is a nice little slide of the 20 most common amino acids. Now, I keep saying most common amino acids because there are a handful of amino acids out there that are being actively studied, and we haven't decided yet if they're common or if they should be included in the list. So don't be surprised if you come across a few more, especially if you do some more research on amino acids, but the 20 that are listed on your screen are the ones that are fully accepted and the ones that are part of the classic models that will make up our proteins. Now within the 20, you're going to notice that they have gone ahead and they've divided some as polar versus non-polar, and it turns out it's an equal division, 10 of each side. And within the polar amino acids, we also see that the side chain can contribute to other factors or other characteristics, such as making the amino acid have more of a negative charge, a positive charge, or a neutral charge. Now, in case you're wondering, no, I'm not going to ask you to memorize these 20 amino acids, and you don't have to memorize if they're polar or non-polar, because that's something that we can easily look up. But I just want you to realize that all amino acids will have their amino, their carboxyl, their hydrogen side, and that the side chain is what sets them apart from each other. So the side chain is what's going to give us our special characteristics that we see featured here, including if it's going to be a polar or non-polar, negative charge, positive charge, or neutral charged amino acid. Once your amino acids are linked together using the peptide bond, what we see happening is that for many proteins, it does not mean that's going to be its final shape. Proteins are well studied for the fact that they have these very elaborate shapes that they will acquire. So what we see happening is that once the peptide bonds are formed, there will be other non-covalent bonds such as hydrogen bonds, van der Waals attractions, and electrostatic attractions that will come in and also play a role in helping the protein fold into its overall final shape. I do want to point out that normally if you're looking at a polypeptide formation, the covalent bonds themselves will form in a very long chain format. That's the picture that we saw previously, right? With the polypeptide backbones just kind of forming units next to each other. This chain tends to be very flexible because your peptide bond will allow for free orientation of the bonds around the open spot of your amino and your carboxyl group. However, what we see happening is that now we can constrain the overall shape by introducing these non-covalent bonds because it's going to allow them to create more of a 3D structure, which is what a lot of proteins rely on to produce their ultimate function. Don't be surprised if when you discuss proteins, you'll come across a statement that will say structure equals function. We've studied a lot of proteins and we see that for many, in order for them to function fully, they need to have a very particular appearance or folding pattern before that function can be adhered to. And it turns out that their ultimate structure will be a combination of the peptide bonds, which link the amino acids together, and then the introduction of these different non-covalent bonds that can interact with different parts of the amino acids to ultimately allow it to fold into a more complex shape. We also know, for instance, that in addition to the three forces or non-covalent bonds that were mentioned previously, so the van der Waals, the hydrogen, the electrostatic, we also know that there are hydrophobic forces that will come into play and will take this freshly made polypeptide chain and will allow it to be folded in a more complex chain. So go ahead and take a look at the illustration that's on the left-hand side. And what we see happening here is the use of hydrophobic forces to make a more complex structure. And these hydrophobic forces are going to allow our polypeptide chain to form in a manner that we're able to separate the nonpolar and the polar side chains from each other. So right over here you can see that the folded conformation, and by the way, conformation just basically indicates the shape. So the folded shape of the protein will allow for the polar sides to freely interact with the hydrogen bonds and water, whereas the nonpolar sides are going to be packed in the core so that they're shielded and they do not have to interact with the hydrophilic components of the aqueous solution that the protein is surrounding itself with. Now, in case you're wondering, oftentimes non-covalent bonds tend to be relatively weak, but what we see happening is that by accumulating them together, so by using several different versions of them within the protein, we are able to form a very strong, solid arrangement. So in case you were wondering, why would we use non-covalent bonds to generate a final protein shape? Because individually they might be weak, but by combining their forces, what we see happening is that they are able to create a very nice, strong bonding arrangement, aka a stable molecule. Another one that I definitely want to point out as well is that hydrogen bonds are a very popular choice within our protein formation and stabilizing of its final form. I know I'd mentioned the hydrogen bonds to you before in one of our previous slides, but I really wanted to include this picture as well because it shows you that you can use a vast amount of hydrogen bonds in different locations. So if you go ahead and take a look at your picture that's on the left-hand side, notice that the hydrogen bonds are given different colorations. The red ones indicate the fact that you have hydrogen bonds that can connect the backbone of one amino acid to each other. The yellow is when the backbone is connecting a side chain of another amino acid. So you can have an interaction between that central carbon and the amino group on a secondary amino acid, or the central carbon and a carboxyl group from a secondary amino acid. You can also get the actual side chains to interact with each other. So now we're taking a look at the carboxyl with the carboxyl, or the carboxyl with the amino, the hydrogen with the side chain, lots of variety. And in fact, there's even been cases, and that's what this little red arrow is trying to show you right over here. There's been cases where you can have that the same side of the amino acid can make multiple hydrogen bonds, meaning that you can have an interaction that will involve the backbone to the backbone to the side chain, and the side chain to the side chain. So the hydrogen bonds are a very popular choice of a non-covalent bond that can be utilized to get the protein to fold into its ultimate shape. Once your polypeptide chain is formed, it turns out that in addition to relying on the non-covalent bonds, as we were chit-chatting about before, we also see that the shape, or I should say the final structure or conformation, is actually determined by largely energetic consideration. The protein wants to fold into a final shape where its free energy is minimized, meaning that it's in an energy favorable format, and it will follow the rules of thermodynamics in the sense of as it starts releasing heat, it will go ahead and contribute to the entropy or disorder of the system. Now, we are going to spend quite a bit of time talking about things like free energy, energy favorable and unfavorable, entropy. We're going to do all that when we start discussing our chapters for our second unit exam, which will involve chapters 3, 13, and 14. So there's more fun to come to kind of get us to learn more about all the different energy shapes and formations within the cell. But for now, what I want to show you is that once the protein has its final shape, it is possible to unfold it, and you can utilize different things, like you can expose it to different temperatures, different pHs, or in this case, in our example, we're exposing the protein to a high concentration of urea, and what we see happening is that it's going to allow the protein to become unfolded. When a protein becomes unfolded, it's because we are disrupting those non-covalent bondings that we're allowing the protein to kind of coil up. And when it unfolds itself, we can often create what we call a denatured protein. A denatured protein, by definition, is a protein that has lost its original shape. Many times a denatured protein, depending on its level of unfolding, can actually completely shut down its function. However, there have been cases where if you remove the item that was unfolding, causing the unfolding, so in this case the urea, the protein can go ahead and refold itself. And as it's refolding itself, we like to say it has renatured itself. So right over here, renaturation requires the correct condition, and it works best for small proteins. Not all proteins will be able to renature. Many of them, once they're denatured, they will lose their function and they will not be able to acquire their natural shape. Now, because we can see that some proteins are able to renature themselves, some research has indicated that their actual instructions for their folding patterns are actually being held within the amino acids itself. However, we have also seen cases that with larger proteins, they might require some assistance from an external source to get to their final shape. And that is then where you come across what we like to call chaperone proteins. So a chaperone protein will often guide newly synthesized polypeptide chains. We see them quite frequently as the end result of transcription and translation, that you now have a beautifully synthesized polypeptide protein, and we see that they are not able to fold themselves. So the chaperone proteins will come in. They will require the use of ATP as well as hydrolysis, so it requires energy to get it done. And most of the living cells will often use chaperone proteins to get to their final shape. Now, the final shape of the protein, I do want to say, when it starts functioning and it starts interacting with other proteins or other molecules, due to the interaction, the shape will slightly change, but it will mostly stay within the same structure level as it was when it was initially formed. Now, our chaperone proteins can go ahead and they can do their work in the cytoplasm of the cell, but we've also seen cases where the chaperone protein will actually sequester the newly formed protein so that it doesn't get a chance to form improperly aggregates or communication with other molecules in the cell. So let's click over to our next slide so we can go ahead and we can take a look at what happens when these chaperone proteins will isolate a new protein from the rest. Here you go. So here's a different look at a chaperone protein. And in this case, the chaperone protein will form almost what they like to call an isolation chamber. And we see that as the newly synthesized proteins come in, they're very simply folded in like maybe a primary or a secondary structure. Well, the proteins will make their way into the isolation chamber, and then there's a little chamber cap that's placed on it. And the neat thing about this is that it allows the peptide to be isolated, allowing it to not make any type of connections with other polypeptides, because keep in mind that the cytoplasm is a very crowded place. We don't want it to interact with other elements because that might cause it to be folded improperly, which will often alter the function of this newly synthesized protein. So the isolation chamber is just basically meant as a way to increase the success rate of allowing your chaperone protein to fold your peptide into its final functioning shape. Once the protein is folded properly, as you can see by the end of our illustration, the chamber cap is released, the protein is ejected out of the isolation chamber, and it is ready to go ahead and do its day-to-day activity. Here is just a look. I love this picture that's on the left-hand side. It shows you the wide variety that you have of these protein shapes. Many proteins, as you guys know, will start off with just a regular ribbon type format, just a linear chain because of the polypeptide backbones. And then what we see is from there on, they can go ahead and they could start coiling around each other like a little helix, or they can make little ribbons like little sheets. And then from there, they can go ahead and cause other protein subunits to gather together so that their size will start to increase. Some proteins are very flexible in their overall format. Others are very rigid. It really all will depend on what their overall function is within the cell. One thing I want you to look at is as you're looking at the beautiful illustration that is on the left-hand side, go ahead and look at the bottom of the slide, and they've indicated for you a size marker. So that way you can see that there is also a wide variety in the size of the individual protein. So for instance, if you were to compare the insulin protein that's figured for us right over here, if we now go ahead and we take a look, for instance, at catalase, or more importantly, remember the hemoglobin we were talking about in our red blood cells? There is just such a wide variety, not only in their structure and their shape, but also their size. And that is then one of the main reasons why when you take a look at proteins, the model that you use to describe them or to illustrate them can vary because it will all depend on what your ultimate goal is to showcase it to your audience. So over here on my second illustration, these are the 4 most common ways that we can go ahead and we can showcase a protein conformation. You can have an emphasis on the backbone. You can go ahead and have what we call a ribbon model. The ribbon model is more of a secondary structure where you're going to take a look at the formation of like the alpha helixes and the beta sheets, which we're going to discuss in just a little bit. You can have a wire model, which will have a heavier emphasis on the non-covalent bonding and the extensions of the side chain. And then you can have a space filling model. And the space filling model will often highlight if there are different subunits of the protein that have to combine together or correspond together to go to your overall shape. All right, now I've mentioned the helix and the sheet several times, so let me go ahead and show you some illustrations so that those of us who are more visually inclined can kind of figure out what I'm discussing. So if you're looking at your polypeptide formation, we talked about the fact that it's going to start off as just a linear chain. And then we see that's its primary structure. And then we see when it goes into its secondary structure, it's going to get on a more complex shape. And it turns out for the secondary structure, the most common folding pattern are either for it to coil up like a ribbon. Let's see if I can make a ribbon. Here we go. That's my little helix. Or to form more of a ribbon structure. Here you go. So this right here, when it's coiling up, making a little spiral helix, that is your helix. And the reason you can make a helix, and you can see it on the picture right here, is what we're going to do is we're going to form hydrogen bonds between the amino and the carboxyl group, but we're going to kind of space them out, and they're basically going to interact. The amino group is going to interact with the carboxyl group about 4 amino acids away. So we're going to kind of space out the reaction. And when it does this, when it starts reacting with side chains, so carboxyl and amino acids, carboxyls and amino groups from between 4 amino acids, when it interacts that way, it creates a ribbon format. It creates a spiral format. So here on your right-hand side in C, you can see this is kind of like the simplified version of what you're doing by forming the hydrogen bonds. And once again, what you're doing is you're alternating the interaction between the amino and the carboxyl group located 4 amino acids away. And that allows it to coil up. On the other hand, what you can do is you can go ahead and allow for the hydrogen bonds to form adjacent to each other, side by side. And when you do that, you get more of a sheet format. And that's what the beta sheet is here for. And when you do your sheet format, it turns out that it can be in what we call an anti-parallel or a parallel format, which I'll show you a little bit more in our next slide. Now, the alpha and the beta were just simply named for the fact that the alpha helix was the first one to be discovered and the beta was the second shape that was discovered, in case you were wondering where did the alpha and the beta come from. Now, speaking of our helixes, just giving you a little bit more detail, we see that the helix can vary in how many amino acids it takes per turn, and we also see that it can vary based on if it's turning more towards the left or towards the right. So on our little illustration right over here, it's showing us 4 scenarios where you have different amounts of subunits, so different amounts of amino acids that are stacked together before you actually have a turning sensation. So for instance, in figure A, we have 2 subunits that are stacked together. In figure B, it looks like we have 3 of them. Yeah. And then in C and D, we have a little bit more because it seems to be a wider turn. So 2, 4, it looks like it's about 6. 6 of them that stack together before you have a full turn, meaning that you're back to the original location of your first repeating subunit. Now, look on the top picture right here, and you can see that depending on how many subunits we put, the overall appearance of the ribbon will obviously either be a tighter turn or a more wide range turn. We also see that the turn itself can either be towards the left hand or towards the right-handed turn. And both of these orientations can be found within the cell, but I do want to definitely point out that the vast majority of our proteins that utilize an alpha helix, they will go ahead and they will highly favor the right-handed turn over the left-handed turn. So this is by all times, this is their favorite, the right-handed turn. Obviously, we can also take our structure and we can see that it can be utilized in many different ways. Like for instance, some alpha helixes will continue to interact until they become a very rigid format, almost like a cord that doesn't have a lot of flexibility to it. These are called coils, and this is, for instance, what you would see in the keratin, the protein that you find in your hair and in your nails. We also see that some of the helixes can start interacting with the phospholipid membrane. So here on our illustration on the side, we can see that our alpha helix is interacting with the phospholipid subunits of a cell membrane, and in that case what we can see happening is that they're able to freely interact with through the hydrogen bonds as well as the hydrophobic components of the cell membrane. Now here's a look at the beta sheets. And the beta sheets are not as flexible as the helixes. They tend to be more of a rigid formation. And as I said to you before, what you're doing here is you're forming your hydrogen bonds, but you're forming them side to side instead of kind of spacing them out like you were doing in the helixes. So you have more a flattening of the protein. And we can see that if we take a look at the orientation from amino group to carboxyl group, if we see that the sheets are running in the same direction from each other, so amino to carboxyl, amino to carboxyl, amino to carboxyl, we like to call that a parallel format. There you go. And if we see that they're running opposite from each other, so amino starts on one end, ends with carboxyl, and then your will have the opposite, start with the carboxyl and end with the amino on the next sheet, then what we're doing is we're looking at an anti-parallel format. Both of these structures are actually pretty common. For the beta sheets, in addition, what we also see happening is that because they have such a rigid format, they will often be used to form the core of many proteins, because the core is in essence so essential for its overall structure properties that need to be built on top of that. We also see that there's a possibility for the beta sheets to kind of stack together and form this insoluble structure called an amyloid. And an amyloid, it almost looks like a zipper. The sheets will intertwine with each other and they'll create a very hard, rigid complex. And oftentimes what we see happening is that the amyloid, depending on its location and its overall size, it can cause some problems within the function of the cells, because it can alter the overall protein shape and it can hinder its function, making the cell not as optimally performing as we would like it to be. In fact, there are some disease models that have been studied that are due to amyloid structures found, especially those found in the brain. Diseases like Huntington's disease, Alzheimer's. Some of you might even have heard of mad cow disease. These are all diseases that have been linked to abnormal amyloid structures being formed. And that basically means that I have a protein that is naturally supposed to form a beta sheet, but it has abnormally stacked on top of each other and created this insoluble, stiff structure that is hindering the cell from performing its day-to-day activity. In fact, let's take a look at this picture right over here. So this one talks about the fact that if you have a misfolded protein, as I mentioned to you before, it can form an amyloid structure. And there are certain diseases that have been linked. Oh, here you go. They mentioned the Alzheimer's. Huntington's disease is another one. Mad cow disease is also. And what ends up happening is that you can have a normal protein shape right here. And when it obtains this abnormal form, what we do is we're going to call it a prion, okay? So if you come across the term prion, that's going to indicate for you a protein that has abnormally folded and is no longer looking the same and also not functioning the same. So that's why on our illustration, notice how they went from circle to more of a square. Now, the problem with the prion, in addition to it no longer functioning, is that there's also been cases that we can see that a prion can start interacting with other proteins that are having their quote-unquote normal structure and shape, and by binding to it, it will actually convert the normal protein to also an abnormal prion form. So what we have now is an ability for the prions to basically start propagating and increasing their numbers. And as they're doing that, as they become larger and larger, they can go ahead and they become amyloid fibrils. And the amyloid fibrils are just literally clusters of these abnormally folded prions that are just taking up room within the cell and the organ. And they're basically interrupting their regular cell-to-cell function. And that then correlates with a lot of the symptoms that we come across with these different diseases that they've been known to cause. So as we kind of get towards the end of our shape and structure analysis, just please keep in mind that the structure of a protein is very complex, but it's also very important for its overall function. And over here, what we're seeing is another example of how a protein can have several layers of organization, and part of that will relate to its intricate shape that it will have for it to ultimately function as we need be. Here we have an example where you have your secondary structure of your alpha and your beta, your helix and your beta, interacting together to create more of a complex tertiary structure. And in that case, what we see happening is that they can go ahead and they can form a domain. Now, a protein molecule can be made out of one domain, but it can also be considerably larger because it can have two or more domains. And what a domain is, it's basically a section within your polypeptide sequence that is able to fold independently from the rest of the chain. So it doesn't have to always fold in the same helical and beta sheet formation. It can have a very distinctive function from the rest of the chain. And you can see that in your illustration right here, you have the yellow section versus the blue section. Both of them belong to the same protein, but they are two separate domains because their folding patterns are completely different from each other. Besides the folding of the protein and the interaction of the amino group and the carboxyl group, we also know that within the protein itself, there will be some unstructured regions. These are usually used to allow for domains to easily connect between each other. We also see that there are polypeptide chains. Many of them will be useful, and part of that is the fact that as the polypeptide chains form, the chain itself will not be utilized for its proper function. It is only when the shape becomes more complex and it goes into the helix, the beta sheet, the tertiary, the quaternary structure, when it becomes more complex in its overall shape, that is then when the true function of the polypeptide / the protein comes to play. We also see that proteins can be classified into what we call families. And for instance, if we take a look at this illustration right here, this is our serine proteases. We can see that they have a collection of different proteins that are slightly different from each other, but more importantly, we see all these different green sections, and these little green sequences indicate areas where they have very similar nucleotide formation. So they have those green areas all in common. And their 3D shape also shares a lot of commonality. And whenever we see a group of proteins that have a very similar nucleotide sequence, a very similar shape, and oftentimes will function in similar ways, we know that they tend to belong to a family. Just like you have your gene families, we can have families of proteins which all originated from an ancestral protein and along the way created slight variations between one and each other. Large proteins are often contain multiple polypeptide chains, and the reason for that is because we see that we can utilize our non-covalent bonds to also bind together protein subunits. And when they combine together in their protein subunits, we can have different ways for this to happen. So for instance, on the illustration on your left-hand side, we can form a dimer. A dimer means that within the binding site, we have an interaction between one and another. So it's only a single identical binding site for each monomer. So that means there's only one other item it can bind to. So then you make a dimer. On the other hand, you can have what we call a tetramer. A tetramer is when you have binding sites that will allow non-identical subunits to interact with each other. Hence the use of the square and the circle to indicate the fact that you are binding non-identical subunits to each other. This all obviously contributes to the wide variety of proteins that we've seen around. And even this picture right here, figure 4.24, this one shows us a protein that is basically using its subunits to form a very symmetrical format. And this is actually hemoglobin. Hemoglobin uses the interaction of 4 chains to form its overall structure, and that is 2 alpha chains and 2 beta chains. And as you can see, if I make a line right in the middle, it is a very symmetrical format with the alpha and the beta mirroring each other on both ends. And this is all due to the interaction of the different subunits. So it's very similar to when we discussed our different binding sites in chapter 2. You know, how many binding sites do the electrons have to interact? It's a very similar concept to the protein. Do you just have one binding site that's very specific? Then you end up going ahead and doing an identical site, which is a dimer. Or you could have some variety of non-identical ones, and then you can create, for instance, a tetramer. It will all be vastly different because, as we've seen by now, proteins come in various shapes and sizes. And here you go. Here's another look at how the subunits are able to interact. As you can see right here with A, if you have a subunit that just has a single binding site, chances are that it will go ahead and assemble into a dimer, because it only has one spot for it to interact with the other subunit. If you have 2 binding sites, as we can see in B and in C, well, then depending on the arrangements of the subunits, we can form a helix or we can form a ring. And then from there on, we can start stacking the subunits and create more of a complex structure, such as the filaments, the spheres, and the hollow tubing. Here is our beautiful actin molecule. This is the thin filament that interacts with the thick filament in the skeletal muscles for muscle contraction. So as you can see, lots of varieties. We have filaments, we have sheets, we have spares, and then also let's go ahead and take a look at the next one, because I want to talk to you about how most proteins, if they have to take up a lot of surface area, they will go ahead and extend in more of a longitudinal shape, versus a globular shape. So let's take a look at that. Here we go. So these are more of an elongated fibro shape. And this is, for instance, what you come across if you take a look at like collagen, or if you take a look at elastic fibers. And it turns out that it will take up more surface area because obviously it's a more elongated molecule that you're looking at. But also notice how the non-covalent bonds will determine if you have more of a stiff, strong structure, or if you have a little bit more flexibility within it. So the collagen one, as you can see, that's all about strength and just creating a tense molecule that's able to withstand trauma or any type of pressure changes. Whereas if you take a look at your elastic fibers, you're going to notice that the non-covalent bonds are basically spaced out in a different format that will allow for a higher degree of flexibility. So you'll have a lot of cross-linkage of these non-covalent bonds that will come into play. Now, obviously, this whole discussion was about the protein shape and structure. So just keep an eye on the fact that all proteins are made out of amino acids. You want to know what the general structure of your amino acids consist of. So the amino group, the carboxyl group, the side chain, and the hydrogen. Please be able to describe the use of the peptide bonds to link together the amino acids, and then the use of the non-covalent bonds that were mentioned throughout, like the hydrogen bonds, the electrostatic forces, to go ahead and allow for the protein to fold into a more complex structure. And at the end of the day, please remember that with the protein, structure equals function. And if we lose that structure, if we denature that protein, chances are that we will lose the function as well. All right, so let's go ahead and let's stop here for part number 1, and we will chat soon, or I guess I'll chat and you'll listen, and we'll take a look at the second part of the chapter, which is going to be how exactly proteins do their work. All right, talk soon.